Welcome to this explainer. Today, we're diving into the secret, shadow world of extra-chromosomal DNA. We're going to be looking at the rogue genetic elements that don't just survive on the periphery, but actually drive evolution, power our cells, and are now being harnessed by scientists to completely rewrite the future of biotechnology.
Here is our roadmap for today. We'll be covering the secret DNA, plasmids, bacterial superpowers, mitochondria, the ancient invaders, chloroplasts, stolen solar power, and finally, harnessing the rebel DNA. Let's just jump right in.
Part 1. The secret DNA. So, think about it for a second. Is all the genetic code that actually drives life securely locked away inside those main chromosomal vaults? When we think of genetics, we almost always picture the nucleus in eukaryotes, right? Or maybe the nucleoid region in bacteria.
Well, actually no. Enter extra-chromosomal DNA. It's essentially a rogue independent operator. It lives completely outside the chromosomes, and amazingly, it replicates on its own schedule. It's usually not even essential for basic day-to-day survival, but it gives cells these absolutely massive evolutionary upgrades. Whether it's floating around in prokaryotes or hiding deep inside the organelles of complex eukaryotic cells, this secret DNA plays an enormous role in powering cells and providing highly beneficial survival traits.
Which brings us to Part 2, plasmids, bacterial superpowers.
In the bacterial world, this rogue DNA acts basically like a deck of genetic trading cards. They take the physical shape of these really small, circular loops of double-stranded DNA. Because they exist totally apart from the main bacterial chromosome, they replicate whenever they want, using highly efficient methods like rolling circular replication to just pump out copies of these extra genes at blistering speeds.
And bacteria actually trade these loops with each other through a process called conjugation to gain instant superpowers. The diversity here is just staggering. You've got F plasmids acting as the bridge builders for the trade. R plasmids, they hand out antibiotic resistance. Col plasmids produce toxic proteins to literally assassinate rival bacteria. And get this, degradative plasmids let bacteria feast on toxic organic wastes like toluene. Then there are virulence plasmids, which can instantly flip a completely harmless strain of E. coli into a severe, dangerous pathogen.
What's wild is that we can actually map these superpower genes with incredible precision now. Take the T plasmid, for instance. It contains dedicated regions for virulence and conjugal transfer that basically act as a biological hacking toolkit. It allows the agrobacterium to inject a piece of its own source code directly into a plant's genome.
And the real-world result of that injection? It's honestly a little eerie. The TI plasmid literally forces the host plant, like the tomato plant, to grow a massive gall. The plant is unwittingly following genetic instructions from the bacteria's extra-chromosomal DNA, producing nutrients that only the invading bacteria can use. It is a stunning, physical example of biological hijacking in action.
Part 3. Mitochondria, the ancient invaders. But bacteria aren't the only ones hoarding secret DNA. Eukaryotic life forms, like us, harness ancient invaders too. Just look at these incredible cellular power plants, the mitochondria and chloroplast. They are membrane-bound organelles with their own distinct circular genomes. That means right now, literally inside your own cells, you have a completely separate set of DNA that does not live in your nucleus. Human mitochondrial DNA is about 16.5 kilobases long and encodes exactly 37 genes, mostly dedicated to running the electron transport chain to pump out ATP. So how did it even get in there? Well, the endosymbiotic theory explains this epic cellular heist in 3 evolutionary steps. Basically, an ancient anaerobic host cell engulfed a bacterium, but instead of digesting its prey, a permanent symbiotic relationship formed. Over time, the host cell actually absorbed many of those bacterial genes right into its own nucleus. Over billions of years, that swallowed alpha proteobacteria evolved into the mitochondria that literally produce the energy required to power your heartbeat today. And because of this ancient bacterial ancestry, mitochondria still replicate semi-autonomously. It looks a whole lot like independent bacteria dividing.
Now, here's a fascinating twist you really need to take away from this. Unlike your chromosomal DNA, which is a mix of both parents, mitochondrial DNA is passed down exclusively from the mother in most animals. So the secret DNA powering your cells right now came entirely from your mother, her mother, and so on. And because it mutates faster than nuclear DNA, we can actually use it as a sort of molecular clock to trace ancient human evolution and ancestry.
Okay, moving on to Part 4, chloroplasts, stolen solar power.
Let's pivot and explore the plant kingdom's version of this ancient invasion. Evolving from engulfed photosynthetic cyanobacteria, chloroplast DNA is actually quite a bit larger than mitochondrial DNA. It organizes into complex circular nucleoids to manage solar power production. Ranging up to 160 kilobases, it's packed with over 100 genes absolutely vital for photosynthesis, including the instructions for critical proteins like ribisco.
But amazingly, the chloroplasts ended up surrendering a lot of its independence over time. Today, a massive 2/3 of its ribosomal proteins are dictated by the host plant's main nucleus. This perfectly illustrates why we call these organelles semi-autonomous. They literally can't survive on their own outside the plant cell anymore because the plant's nucleus has stepped in and taken over manufacturing most of the parts they need to function. Nature, of course, takes this hijacking to some totally bizarre extremes. Meet the leaf sheep slug. It literally eats algae, steals their chloroplasts, and keeps them alive inside its own tissues to photosynthesize. It's a transient endosymbiosis known as kleptoplasty, basically turning the slug into a solar-powered animal. Just a striking, incredible example of how life continues to trade and hijack these extra-chromosomal organelles.
Finally, let's wrap things up with Part 5, Harnessing the Rebel DNA.
By understanding these genetic trading cards in ancient power plants, we are driving modern biotechnology forward. We're talking everything from CRISPR gene editing tools to creating transplastomic plants that can produce edible vaccines. And because chloroplast DNA is strictly maternally inherited, modifying these crops means the engineered genes won't blow away on the pollen and accidentally cross-contaminate wild species. Plus, we're actively using bacterial plasmids every single day as standard cloning vectors to deliver synthetic biology and targeted gene therapies.
When you look at the big picture side by side, the contrast is just amazing. On one end of the spectrum, you have these tiny, fully autonomous plasmids trading superpowers horizontally across bacteria, and on the other, you have massive, semi-autonomous organelles, passed down strictly maternally, acting as the permanent power plants for complex life. Which brings us to a final, provocative thought to close out this explainer. Now that we possess the ability to actually engineer this rebel DNA, what extraordinary leaps are we going to make next in human-directed evolution? From endosymbiosis billions of years ago to CRISPR in the lab today, extra-chromosomal DNA proves that sometimes the most powerful secrets aren't inside the vault at all. They're hiding in the shadows. Thanks for learning with us today.